Alrighty. Okay, last person voted and poll. Need to just do some sharing. All right, looking good for where we are. So I think always a great reminder to go back and review what we've been learning, staying on top of that material as we're adding new layers. What you're going to see is or hopefully you're noticing, as we learn one concept on Tuesday, we're oftentimes applying it and building on it on Thursday. So making sure you're staying on top of the material is really important. Otherwise, it's really easy to get lost. So that being said, yes, the bulk of you guys are on the right track. Exocytosis is a form of bulk transport, and remember that that is a type of active transport. It requires direct investment of ATP energy in order to move a large amount or a large item across the plasma membrane. So diffusion, osmosis, filtration are all examples of types of membrane transport that utilize other types of energy. So we talked about how diffusion and osmosis utilize kinetic energy, whereas filtration utilizes pressure. So blood pressure in this case, in the case of our body, I should say. So make sure you're staying on top of that material. If you didn't get this question right, you might want to go back and review those concepts. If you did get the question right, awesome, you're on the right track. Good job. All right, so we left off talking about energy. So today's going to be, I think, we're going to rip the Band-Aid off of the, cellular respiration and photosynthesis lectures. Again, I'm not a cell biologist, so not my favorite lectures of the semester. But I do appreciate cellular respiration quite a bit, and I love the electron transport chain. So I'll talk a little bit about that to start off with, some of the building blocks of the chemical processes that are occurring for both cellular respiration and photosynthesis, and I'm hoping that we will get through at least cellular respiration today, if not both cellular respiration and photosynthesis. All right, so we left off recapping how law of conservation of energy is something we learned about the very beginning of the semester. Energy is not created, it's not destroyed, it's just converted from one form to another. The first law of thermodynamics, we already talked about that. Second law, we're adding today, and we're going to be talking about how those energy conversions are inefficient. Every time you convert energy from one form to another, you're going to lose some of that energy as heat. It kind of reminds me of like when you exchange currency, you always lose a little bit of your value to fees. Energy conversions are inefficient. And we're going to see that that's going to happen quite a bit. And over the course of evolution, critters have figured out a way to take advantage and make use of that lost energy. So every time we cycle ATP, say, we're going to lose some energy as heat, but that is the heat that you use to keep your body warm, right? So we're taking advantage of that energy loss and using it for something else, right? To maintain constant environmental temperatures. Hey, I hear you. I didn't put him on tie down this morning, and he's off in the bedroom doing naughty things. All right, adenosine triphosphate. I've been talking about this molecule since probably the first week of class. ATP is the abbreviation, right? So it's got an adenosine backbone with a little ribose sugar, and then it's got this three phosphate groups tacked onto the end here, right? So here you can see the 1, 2, 3 little phosphate groups. And ATP is the universal currency that we use in our cells to fuel work, right? So it is the energy for all living organisms are using ATP. Bacteria, so our prokaryotic cells and eukaryotic cells are using ATP. Really interesting, I think, molecule. So let's talk a little bit about what makes it interesting and why it evolved to be the most universal used cellular energy. It has to do with the bonds between these phosphate groups. They're oftentimes referred to as high energy phosphate bonds or high energy bonds. And that's because they have this crazy property where they're oftentimes described as a loaded spring. right? Which means if you've ever had that experience where, I don't know, fiddling around with your pen and it breaks and it has a little spring inside. Yeah, you guys know what I'm talking about. When you compress the spring, right, it's kind of like the equivalent of putting this phosphate bond on, right? Attaching a phosphate, right? It attaches and you've stored some energy in that bond. But the thing about this high energy bond is that when you break the bond, remember when we break chemical bonds, we pop electrons out of the orbital, we harvest that kinetic energy to utilize that energy. In the case of this phosphate bond, when you break the bond, you pop the electrons out of the orbital. But the properties of this bond also cause a little extra release of energy. So it's like disattaching your fingers from that spring and having it, boom, bounce back, right? It gives you a little bit extra release of energy. So that phosphate bond, because it gives you that little bit of extra energy, turns out to be a really efficient molecule to use for cycling energy in your cells because it is very powerful when you break that bond. Now, here's the thing about this high energy bond that's, I think, the most interesting part about ATP is that it's unstable, right? It's kind of like, ooh, ready to pop, right? And ready to release its energy. And so it's really unstable, which means, right, you can't store it, right? So you can't build up ATP in your cells for a rainy day. You cannot store it. You have to make it on demand, right? And so that's going to be really impactful to the process of cellular respiration. And the beauty of the stages of cellular respiration is the fact that we can cycle ATP and store it, right? So I'll come back and talk about that property in a little bit. But when you break that phosphate group off, right, you have created or removed the high potential energy status of that ATP molecule, and you've converted it into what we call ADP, adenosine diphosphate. Notice how it has only 2 phosphate groups, and then the extra little phosphate that's now floating around in the cell. Phosphate groups we use quite a bit for all kinds of different jobs. I'm not going to talk too much about phosphorylation, but if you go on to take physiology, you'll learn all about how phosphate groups are really important for a lot of the chemical reactions that we have in our body. All right. Does this make sense so far? So I want to remind you what we talked about. Did I talk about this? Yes, I did talk about the snowboarder. I want to remind you what we talked about last class. The high potential energy status, right, is when you have energy available for work. You can utilize that energy, convert it into kinetic energy, right? And then once you've released all the kinetic energy, you end up in a low potential state. You know, like snowboarder at the bottom of the hill has low potential energy. You have to invest some energy in order to restore the high potential energy status. And you're constantly going to be cycling between high potential energy and low potential energy all the time, right? As you cycle energy. And ATP is no different, right? It's the same thing as that snowboarder. ATP, the triphosphate version, is the high potential state. It is like a snowboarder at the top of a mountain, right? Lots of potential energy. And when we pop the electron out of its orbital and break that phosphate group out, we release that kinetic energy and harvest it to do work. And then we end up with an ADP and that phosphate group that's now disattached, low potential energy state. And what we're going to have to do is invest a little bit of energy, right, to reattach that phosphate group and cycle ATP again. Right. So, and again, remember that high energy bond is basically what it means is the energy that you invest to attach the phosphate, you get more when it's broken, right? Because of that special type of bond that it is. So you release more than what you invested in reassembling that ATP molecule. So it's a win, cellular win. Does this make sense? ATP cycling? Remember that if you're not cycling ATP, that means your cells are dead. Yeah, you can't be alive if you're not cycling ATP. Really important process. So, what do we use ATP energy for? All kinds of different jobs that we're doing in the cell. So last class we talked about beating cilia and how you're literally moving those cellular extensions, or beating flagella, right? You're literally moving those structures. This is called mechanical work, right? That costs ATP energy, right? It's not going to happen just on its own. Transport work. We talked about this already as well. So if we're transporting something across the membrane, if we're moving it against its concentration gradient, or if we're moving a lot of something or a big bulky molecule, it's going to take ATP energy. This is one of the places where we're using that ATP all the time. Chemical work, right? Remember we said that breaking bonds requires a little bit of activation energy. Right? Some chemical reactions are going to require a fair amount of energy. They're called endergonic reactions, right? Reactions that require an energy investment in order to run, right? So for that, we're going to be using ATP energy. And then we'll see your body's really amazing at cutting costs, right? So you oftentimes see that reactions are going to be coupled with each other. Some chemical reactions are actually going to be exogonic, where they release energy. And what that means is that you can now couple up a reaction that takes energy with a reaction that produces energy, and you end up with a net not having to invest as much to get the reactions to happen. Does that make sense? Multitasking, in other words. Yeah. All right. Okay, so really quickly, chemical reaction categories. We've got catabolic pathways, which is basically a fancy term that means this is a chemical reaction that's breaking apart molecules, say, right? So you're going to be running hydrolysis, for example, is a catabolic pathway. Right? So for cellular respiration that we're going to talk about quite a bit, it's a catabolic pathway. You are breaking down a sugar monosaccharide to release the energy that's stored in those bonds. Versus anabolic, which is, I always think A for assembling, right? It is what's called biosynthetic. It's making some kind of biological molecule. Right? So for example, when we talk about your ribosomes assembling protein strands from amino acid monomers, that's an anabolic pathway. So anabolic pathways create something, right? Catabolic pathways break them down, right? So another way to think about them is catabolic pathways are hydrolysis reactions, right? Anabolic pathways, therefore, are dehydration synthesis pathways, right? When you're building molecules. You thought you could data dump all that stuff from last unit. Turns out we're going to keep using it. Make sense? Categories of chemical reactions? All right. All right, and then we've talked about this a couple times already, the fact that metabolism is a series of chemical reactions. So when you're talking about your metabolism in your body, you're talking about the chemical reactions that are running to keep you alive and to maintain homeostasis inside of your body. And so here's the chemical reaction that we're going to spend a lot of time learning about today, cellular respiration. So one of the things that I'm going to point out and we're going to talk about is that these are pathways, right, that incorporate or include a bunch of chemical reactions. It's not just magic, all of a sudden that glucose is going to break apart, right, and you're going to release and cycle ATP. There's a whole series of chemical reactions that happen from the process of converting your reactants into your products. And they're going to require ATP energy, and you're going to be using all kinds of different enzymes to get those reactions to occur. If you go on to take cell biology, you're going to learn all the enzymes, every single reaction, every cofactor that's used. It's so exciting. For us, we're going to break it down into just 4 of the major steps, right? So, phew, right, intro bio level of understanding cellular respiration. Okay, last thing. Hey, I hear you. Last thing I'm going to talk about here is the reduction and oxidative reactions that we're going to be talking about, right? So oftentimes if you've taken chemistry, you refer to them lovingly or hatingly, I don't know, redox reactions. where you're going to be having one molecule being reduced and another being oxidated. And so I don't want to spend a ton of time here, but I do want you to know these two terms, the reduction and the oxidation, and understand what they mean. So again, we're going to be building on our chemistry foundation. So when we talk about a reduction, right, you're talking about the molecule that has gained an electron. And I don't know, in my crazy way of like processing information when I was in college, right? If you're gaining an electron, which is a negative charge, you're being reduced, yeah, because you're going negative. I don't know if that works. Whereas oxidation is going to be the molecule that loses the electron. So anytime you take an electron from one molecule, it's going to end up going to another molecule, right? So these oxidations and reductions are oftentimes paired, right? So in chemical reactions like cellular respiration, notice how the sugar is going to be oxidated, right? It's going to lose an electron. And that electron, we're going to talk about that electron a lot, right? We're going to use it to run something called the electron transport chain. Right? It's going to be one of the ways that we harvest and store energy to use to cycle ATP, right? And then whoever receives that electron, in the case of cellular respiration, it's going to be oxygen, right? And we already know this. Oxygen is electron greedy, so it's more than happy to take the electrons that are floating around in the environment or steal electrons from a chemical reaction. It's going to be reduced. So if this helps out, there's a little mnemonic, oil rig, which stands for oxidation is loss, reduction is gain. So oil rigs like off the coast that are drilling for oil, maybe that'll help you out. Good? All right.
Can you say that again? Oxidation is lost, reduction is gained. Got it.
You got it. Even just speaking it out loud, right, reinforces that information. So I don't know, even if you guys just want to go back and, read out loud to yourself your notes, it helps out in the learning process. All right, so here's the beautiful thing about electrons and transfer of electrons is, when oxygen is stealing an electron, it doesn't just go, oh, may I please, sir, have your electron? Why, thank you very much. No, oxygen is not polite when it is stealing electrons, right? It reaches and grabs, right, and takes that electron. It's a powerful move, right? So in that analogy, what happens is when oxygen takes an electron, it's a boom, big release of energy, right? And it can be a big release of heat or light energy that happens, right? Unless you have a way of capturing that energy, right? So when oxygen steals an electron, it's a poof, big release of energy. Freak the dog out. All right, make sense? Big release of energy. So, right, we know this in our everyday life, right? Because if you are 21 and you go out to celebrate your birthday or whatever, and you get one of these fancy cocktails that's lit on fire, right? What's happening there, right? Oxidation. Oxygen from the atmosphere is stealing electrons from the sugar that's in your alcohol, right? And because there's nothing there to capture the energy, all that energy, as oxygen is ripping electrons away, is released as heat and light, fire. Yeah? So when you're at the campfire, right, and you're burning logs, right, oxygen from the atmosphere, right, is stealing electrons from that wood. And in the process, releasing light and heat energy. Right? This is why you constantly have to stoke the fire to make sure oxygen's flowing through there to keep it happening. Eh? Oh, oxidation's kind of cool. All right, good. Releases a lot of energy and power. If we go back to our high potential energy, right? Example of storing water behind the dam, right? And then harvesting it by kinetic energy. If we relate this to what we just talked about for oxygen stealing energy, right? It would be like, oh, a molecule having an electron ready to be stolen to release energy is all this water behind the dam. Oxygen strategy would be to bomb the dam and break it apart, right? And all the water got released at once, right? That wouldn't allow us a controlled, efficient way to harvest energy, right? We don't do that when we're releasing water from a dam to harvest energy. We release a little bit at a time, right? So that we can more efficiently harvest that energy. Yeah? I'm building towards something here. Just bear with me. All right, so here's my crazy Mexican heritage example of how to understand why this process is so important, right? So I don't know, when I was a kid, we used to go to birthday parties. God, it seemed like almost every weekend, right? And there was always a piñata, right? You guys done a piñata before, right? As kids, right? They're not filled with little jiggers of booze. They're filled with candy, right? And invariably, right, there was the one kid that like blew the piñata, and moms were all really upset, right? Because the piñata was the way to keep the kids distracted and they get to take a break, right? And so, you know, good moms put the powerful, biggest kid last, right? So that you don't destroy the piñata, right? You can get some time out of that. So when you have the big kid, pow, the piñata, right? All the candy gets released at once. Big explosion of energy being released, right? And if you're one of the kids standing there, you can grab a handful, but imagine you've just broken this piñata over a grate, right? So you grab your handful of candy, you put it, your candy energy in your pocket, and by the time you go to reach to grab another handful of that energy that's being released, all the energy is falling through the grate. Right? Not very efficient. Right? So how am I going to slow this process down so that I can take multiple handfuls, right, of candy energy, right? Or energy being released when oxygen steals an electron? Right? For that controlled harvesting of energy, we're going to be using something called the electron transport chain. This is the part I really love, right? This blows my mind that this happens. So electron transport chain is going to allow you to slow down that movement, that oxidation of sugar and the reduction of oxygen, right? It's going to allow you to harvest that energy in a controlled, stepwise fashion so that you can pull as much energy out of that release, that big, powerful release of energy as possible. It increases efficiency. And indeed, it's a really evolutionary conserved process. We see it across the board. All living organisms use this process. And we use this process, the electron transport chain, for a bunch of different metabolic pathways, right? So for us, we're going to talk about, we use electron transport chain for cellular respiration. It's also used for photosynthesis as a way of harvesting energy in a controlled fashion. Right? So really important, electron transport chain. So let's talk about how it works. Are you guys ready? And this, I think, can be kind of mind-boggling to imagine this is happening inside your mitochondria. So remember that inside your mitochondria or inside your chloroplast, I mentioned how there's folds of phospholipid membrane, right? And in those folds of membrane, right? So here in this, really, I don't know why I keep using this funky illustration, but it's a really grossly oversimplified illustration of the electron transport chain. And notice how you're inside of the mitochondria. And here's one of those folds of what we call cristae. This gray here represents phospholipid bilayer. And all these purple and magenta dots are representing the big purple are proteins, right? And then the magenta are some of the cofactors that you're going to be using to help move the electron away. And again, we're not going to get into each step and naming each one of these items. That you get to do if you go take cell bio, right? For us, we're just going to say it's a series of membrane proteins, right? And what we're going to see is that we're going to take the electron off of the sugar, and we're going to temporarily store it in this NADH molecule. And I'll come back and talk about this in a bit, but for right now, I want to talk about the electron that we're harvesting. So we pull that electron off of the sugar. We use NADH to transport it over to the electron transport chain and temporarily store it. And then when we're ready to run the electron transport chain, right, we're going to pop a hydrogen off of that NADH and steal the electron associated with that hydrogen. So we're going to leave an H plus. Yeah? Okay. The electron, I'm going to do a molecular hot potato. You guys know the hot potato game, right? Where you're tossing. In this case, these proteins and cofactors are playing hot potato with the electron. Each molecule or protein is more and more electronegative to drive this stealing of the electron. I'm not going to get too much into that. Hot potato with the electron means that every time you toss it, that means you're popping an electron out of its orbital. And remember that releases kinetic energy that you can then harvest to do work. Right? So by popping the electron again and again between all these different proteins and cofactors, you get to, instead of taking the electron and putting it directly on oxygen, you detour through the electron transport chain so that you can control that harvesting of energy. So this is the way we slow down that reduction so that we can harvest as much energy as possible. And what are we going to do with that energy? We're going to use, every time we pop an electron out of its orbital and release kinetic energy, we're going to use it to pump hydrogen to one side of the plasma membrane, or the phospholipid membrane, I should say. Right. So here you can see what you're doing is building this hydrogen ion gradient. Remember that at the cellular level, that's like storing water behind a dam. It's storing potential energy. Right. So you're taking the energy that came from pulling that electron off of the sugar and transferring it to oxygen, big release of energy. I'm slowing it down and controlling the harvest of that energy by running the electron through the electron transport chain before I then at the end give the electron to oxygen. I'm using that energy to pump hydrogen ions from one side to another to store this proton gradient. So it's sometimes called, these hydrogen cations are sometimes called just protons. Yeah, it's like storing water behind the dam. Why do I do this? Remember what we said about ATP. ATP is not stable. I can't store it. Hydrogen gradients like this, I can store all day. Right? So this is how I'm storing energy to have it ready to go when I'm ready to cycle ATP. Good?
I have a question, Professor.
Yes.
So the transport chain, did you say that it's creating the hydrogen?
It's pumping the hydrogen.
Hydrogen. Okay. Thanks.
Yeah, I'll add a little bit more layers of detail to hopefully help understand this process, but I'm gonna slowly, incrementally add little bits of detail and repeat a lot because I know this can be a hard concept. So let's watch the little animation here. So here came in the NADH. We pull off the H and harvest the electron. And notice how in this case it's going down all these really weird, funky, right, membrane proteins. The yellow represents the phospholipids. And notice how when we get to key stages, we use that energy to pump hydrogen from one side to the other. Right? That phospholipids making a barrier. The ATP synthase part is a different step. I'll get to that in a bit. So let me go back and we can watch it one more time. Oops, maybe not. So here's the phospholipid membrane. Here's all the membrane proteins and cofactors that make up your electron transport chain. We're going to talk more about NADH in a bit, but we're going to take the hydrogen and the electron off of the NADH. We're going to pop the electron down, 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 down the electron transport chain. As we're popping electron from one factor to another, we're using that energy that's harvested to pump hydrogen from one side of a plasma membrane or one side of a phospholipid membrane to another. Yes? I should put a little bit over that. Electron transport chain. Ooh, allows me to store energy. Again, that hydrogen gradient is like storing water behind a dam. Okay? When we are ready to cycle ATP, then we can open the dam doors. Always makes me giggle. All right. Good. Same thing. Same principle, I guess. Oh, so exciting. All right. Last step. How do I get from that hydrogen gradient to cycling ATP? So this stage is called oxidative phosphorylation or chemiosmosis. Right? Both mean the same thing. All right. So at the end of my electron transport chain, I am have stored quite a bit of potential energy in this hydrogen gradient. When I am ready, when I need ATP, right, then I'm going to open the dam door. And the dam door is this membrane protein that acts as an enzyme. It's called ATP synthase, right? It's going to open, and it's permeable to those hydrogen ions. So hydrogen is going to naturally rush or diffuse down its concentration gradient through the ATP synthase. And ATP synthase literally churns and spins in that phospholipid membrane like a turbine that you would have at the dam, right? And that energy, right, that kinetic energy is going to be used to attach a phosphate group back onto ADP. Use that energy to restore the high ATP energy state. Chemiosmosis or oxidative phosphorylation. Isn't that beautiful? Right. So the hydrogen gradient can persist, right? Remember ATP energy, ATP molecules are not stable. You have to make them on demand. So say I decide to get off the couch and all of a sudden my muscles need more ATP. They open ATP synthase and let it harvest energy to cycle ATP. We're doing this all the time, right? Pumping and storing hydrogen gradients, and then using those hydrogen gradients to cycle ATP as you need it. And I don't know if I mentioned, right? You cycle your body's weight worth of ATP every day. It's stunning, right? Once you use that ATP, right? You're going to break that high phosphate bond. Remember that this is how you release energy to do cellular work, and that's going to generate heat, right? And that's how you keep your body warm, right? Also, coincidentally, right, when you're working out, guess what happens to your ATP needs? Up or down? Up. Right? Because you're using your muscles more. Muscles are really expensive. They use a lot of ATP. Your skeletal muscle cells have your highest concentration of mitochondria of any cell in your body. Also because they happen to be pretty big. But as you're cycling more ATP during working out, right, you're generating more heat. This is what makes you hot when you're being active, right? The fact that you're cycling more ATP and releasing more heat energy. Cool. So ATP synthase is literally, right, a membrane protein, right, that is residing in the phospholipid membrane folds inside of the mitochondria. Kind of fun. Any questions? Electron transport chain and chemiosmosis?
So the more heat energy you create, then that's why you sweat when you're working out?
Mm-hmm. so that
Yes.
you can evaporatively cool and release that extra heat.
Got it.
Oh, biology. Look at how much you guys have learned. All right, any questions on this? Okay, so here's the good news. Oh yeah, I want to say that. The good news is chemiosmosis or oxidative phosphorylation and electron transport chain are two of the 4 steps of cellular respiration that we're going to be learning, right? So you're halfway through cellular respiration. You didn't even realize we were talking about it. So let's do this. Here's what we're going to move on and talk a little bit about. My computer zooming. Sorry. Okay. So let me give you some context to cellular respiration and photosynthesis, these 2 processes, and then we can focus in on cellular respiration. I actually think we might be able to get through cellular respiration and photosynthesis today. 2 critical metabolic pathways for life on Earth, changed life on Earth. These 2 pathways. So pretty darn important. And remember, they are both going to be using electron transport chain and chemiosmosis or oxidative phosphorylation. They both use that process. So for cellular respiration and photosynthesis, I always joke that they go like this in terms of regulating life on Earth, right? So we're going to talk a little bit about energy flow terminology at an ecosystem biosphere level. We're going to talk about the relationship between photosynthesis and cellular respiration, why they're like this. And then I'm going to spend most of our time talking about cellular respiration. We'll talk a little bit about the terminology. You're going to need to be familiar with the metabolic pathway, the chemical equation. And then I'm going to talk about the 1, 2, 3, 4 steps, right? 4 major steps. Keep in mind that there's over 20 chemical reactions that happen. You're only learning 4 of the steps. And in fact, you already learned electron transport chain and chemiosmosis, right? So we'll add glycolysis and Krebs cycle to that. So you're going to need to know each of the 4 steps, right? The order that they occur and what happens during each step. So I'm going to add a little bit more detail. We're going to talk a little bit about what NADH is and how it works. And then I'm also going to talk about, I don't know what happened to my bullet here, anaerobic cellular respiration, because not all cellular respiration is aerobic. So I'll mention anaerobic at the end there. All right. And keep in mind that the 2 reactions are pretty immersed with each other, right? When we take a look at photosynthesis a little bit later, remember that prokaryotes evolved photosynthesis early on in, maybe after 1 billion years of life on Earth. And that was really important, right? Because photosynthesis allowed those early prokaryotes to pull carbon dioxide and water from the environment, harvest sunlight energy, and convert it into sugar energy. And the byproduct is oxygen, right? And when we take a look at that, it turns out that the products of photosynthesis are the reactants of cellular respiration. So then other prokaryotes are going to turn around and use that glucose and oxygen for aerobic cellular respiration to cycle ATP. And as they break down that sugar, they're going to chop it back down to the carbon dioxide and water, right? So in other words, right, photosynthesis is the anabolic building. Cellular respiration is the catabolic pathway, breaking it down. So you're building sugar, breaking it down, building sugar, breaking it down, building sugar, breaking it down. Right? And super important, right, in a relationship between each other. And indeed, right, that anaerobic bacteria or that aerobic purple bacteria lineage eventually led to the mitochondria, and the cyanobacteria that invented this process eventually led to the chloroplast of a eukaryotic cell. So really important. I could talk a lot more about that relationship, but not the context of our lecture. So how do we describe organisms that are using these different pathways? We're going to start to introduce a few terms in terms of how organisms acquire their energy. So photosynthetic organisms are autotrophs, and autotroph literally translates to self-feeder, right? They're able to harvest sunlight energy in some ecosystems, other types of chemical energy to make their own organic molecules, right? In other words, they don't have to eat somebody else in order to get their monomers. They can make their own monomers. A heterotroph is an other eater, right? Other eater, right? And heterotrophs are going to have to get their energy by ingestion. They're going to have to eat. They're going to have to consume, right? Another organism or other organic material in order to get their monomers. They can't make their own monomers. So plants are, and I keep defaulting to saying plants, but remember, plants are not the only autotrophs out there. There are also some unicellular eukaryotes, what we collectively used to call protists, right? That are also autotrophs. Photosynthetic bacteria are also autotrophs. So cyanobacteria are autotrophs. The big difference there is that they don't have a chloroplast because they're a prokaryotic cell. They don't contain membrane bound organelles. Does that make sense? Hopefully. All right. So how we get sugar by photosynthesis, right? If you're an autotroph. If you are a heterotroph, you have to consume some type of autotroph or other organism to get the sugars that you need to run cellular respiration. The other thing we want to remember and keep in context is that you guys all know we have to breathe in order to stay alive. And if you don't breathe, you die. Yeah, we know this. Why is breathing important? Remember that every time you're breathing, you're inhaling atmosphere that's rich in oxygen, right? You're acquiring that oxygen. Every time you exhale, you're releasing carbon dioxide. Right? It turns out that those are going to be completely related to cellular respiration that's happening in your cells. That oxygen that you acquire in your respiratory system is going to be delivered to every single cell in your body, right? And those cells need oxygen in order to run cellular respiration and cycle ATP to stay alive. Right? And as they cycle ATP, right, they chop that sugar back down into carbon dioxide, which would, if it accumulated in your body, disrupt your pH. So you have to get rid of it. So it diffuses into your bloodstream, and your respiratory system allows you to get rid of that carbon dioxide every time you exhale. So breathing is fundamental to being able to run cellular respiration. If you're not breathing, then you're not supplying your cellular respiration. You can't cycle ATP and you can't survive. Yeah? Cool. All right. All righty. Eating is also important, you know, right? If you don't eat, you die. Why? Right? Because this is how you're getting monomers, right? You're getting the monomer, the macromolecules that you're going to break down or convert to glucose. And glucose is fundamental. It is the sugar source that you're going to be using to run cellular respiration. If you are not getting enough nutrients by eating, right, you may not have enough glucose to supply your cells to cycle ATP. Causes, it's a little bit more indirect, but causes lots of problems. Right? And the way that you're going to acquire that is your digestive system. Right? So your digestive system, your respiratory system, your cardiovascular system are all supporting cellular respiration. Yeah? Ooh, all ties together. Scale. All right. So we mentioned that cellular respiration is aerobic or there can be anaerobic forms of cellular respiration. Bacteria have been very creative over the course of evolution, and they have all kinds of different versions of cellular respiration, right? They're going to yield different waste products and different amounts of energy. I'm just going to oversimplify and talk about the anaerobic pathways that yield only a little bit of ATP, maybe 2 ATP. But there are some intermediates. Again, microbiome, I think microbiome, you'll talk all about that, the different forms of fermentation. So aerobic, we're going to learn, is going to utilize oxygen. It is going to yield the highest amount of ATP per sugar molecule. So when aerobic cellular respiration evolved, thank you, purple bacteria, it revolutionized life on Earth. I'll talk about that in a bit. All right, so here's what I want you to pull out of this particular slide, is when you consume food, if you're a heterotroph, right, you're going to break it down using enzymes in your digestive system. It'll get absorbed and delivered to your cells. Your cells are going to be receiving glucose, that monosaccharide, that's then going to be used in cellular respiration. The first step of cellular respiration we're going to learn is glycolysis, right? Lysis, do you guys remember this word root? It's what happens to animal cells to break. Yeah, is that what you said?
Yeah.
I can't hear over the dog ripping paper in the corner over there. Such a naughty dog. Hey! Lysis means to break or rupture. Glyco is referring to that glucose, right? So glycolysis is literally means sugar breaking, right? You're going to break the sugar apart. And then those broken bits can be used either in aerobic cellular respiration or anaerobic. The point I'm trying to make here is glycolysis, this first step, is going to be universal to all types of cellular respiration. Right? And then anaerobic, I'll come back and talk about at the end. This is when we're not using oxygen, and there's a lot of different pathways there. And I'll talk about that more later. Remember that we're looking at cellular respiration. Hey, that is a oxidative reductive reaction. Remember that we're going to be taking electrons or oxidating glucose, right? Eventually, those electrons are going to head to the oxygen, right? The oxygen is going to be reduced, right? And as we chop up, right? So if we take a look, as we chop up the sugar to its component parts, we're going to create CO2 or release CO2 and water, right? So we're going to get, as we chop apart this 6 carbon sugar, we're going to yield carbon dioxide, and the hydrogen we're going to use in the electron transport chain, and some of it's going to combine up with some of the oxygen to make water. So we're breaking apart the sugar, right, to release the energy in those bonds. Right? The sugar is going to be oxidated. It's going to lose its electron. The electron that we take from the sugar is going to run through the electron transport chain and eventually end up at oxygen. Right? But again, we're going to use the electron transport chain to slow that release of energy, right, and harvest. So should you be familiar with this chemical reaction? This is a reaction for aerobic cellular respiration. We're going to take sugar and chop it up into pieces, right? We're going to be using oxygen for its oxidative powers to steal that electron, right? And release energy. Right? When we break apart that sugar, we're going to yield CO2 and water and cycle ATP energy with the energy that's released by transferring the electrons over to oxygen. So I'll talk about all these steps as we go through. So remember, aerobic cellular respiration has over 24 steps, lots of different chemical reactions. We're going to break it down into 4 major events, right? And I'm going to remind you that all of this is happening inside of the mitochondria, right? And remember that inside of your mitochondria, you've got all these folds of phospholipid bilayer, right? Your electron transport chain, those little membrane proteins, are going to be studded, right, all over the folds of the cristae, right? The more folds you have, the more electron transport chains you can support, right? And so if you take a look at the folds, right, they're going to allow you to pump hydrogen from this side to this side and make a little gradient, right, all throughout the cristae. I'm really bummed that we're not in the classroom because we've got this really great teaching mural in the bio lab where we have a fabulous illustration of the mitochondria. I should maybe take pictures since we're going to be online in the spring as well. All right. So what are your 4 steps of aerobic cellular respiration? Glycolysis, Krebs cycle, electron transport chain, and chemiosmosis. And again, we already talked about 2. So I'll come back and review these guys as we get through in case it didn't stick the first go round. But let's learn a little bit more about glycolysis, that first step that's breaking down the sugar. So again, you need to know the 4 steps. You need to know what happens in each of the 4 steps, right? And you need to know what's yielded from each of the 4 steps. So this is what we're going to talk about next. All right, so glycolysis, first step. You bring your sugar into your cell, right? And it turns out that glycolysis is the only step that's not going to happen, right, inside the mitochondria. Happens in the cytoplasm, right? So glycolysis, we're going to take that 1, 2, 3, 4, 5, 6 carbon sugar, and we're going to break it in half. And notice how we're going to yield 2 3 carbon pieces. Right? Those 3 carbon pieces are going to be pyruvic acid. Right? So if we break this bond during glycolysis, we're going to yield 2 of these pyruvic acids. They'll be edited a little bit by some enzymes and converted into pyruvate. Well, they'll get edited and then enter into the mitochondria. Right? So the rest of the steps are going to happen inside the mitochondria. So glycolysis is unique in that it takes place in the cytoplasm. And then the 2 3 carbon sugars head into the mitochondria. Now, here's the thing about glycolysis. That sugar molecule is stable. It's stable. It doesn't want to break. So we're going to have to use enzymes to get to motivate to break. And we're going to have to invest 2 ATP worth of energy in order to get glycolysis to happen. Right? So glycolysis is oftentimes referred to as the energy investment stage. Hey, no, is oftentimes called the energy investment stage because you have to invest 2 ATP to get it rolling. Right. Now, every time we break bonds, we can cycle ATP. There's multiple ways to cycle ATP, and I'm not going to get into the details, but oxidative phosphorylation or chemiosmosis is how you're going to be able to be the most efficient, right? But you will get a few ATP using other processes as we go through, right? So notice how glycolysis is going to produce 2 ATP, right? It uses 2, it produces 2, so it's a net 0 gain, right? It's also going to yield 2 NADH. And so NADH, I pointed out, was that starter molecule for the electron transport chain. What I want you to know about NADH is I just want you to think about it as a temporary energy storage molecule. It's going to be picking up the electron from the sugar, right? And temporarily storing it until you can deliver it to the electron transport chain. It's a temporary storage molecule. Delivering hydrogen, right, and electrons. All right. Good with glycolysis? 2 NADH, 2 ATP, but it required 2 ATP. Right? Happens in the cytoplasm. Breaking the sugar from a 6 carbon piece to 2 3 carbon pieces. I left my pop beads at school too. All right, those 2 3 carbon pieces, they get converted into different types of molecules as they're being shuttled into the mitochondria, right? Turns out that the 2 pyruvic acids are not permeable to that mitochondrial membrane, so they have to be converted. I'm not going to talk about those intermediate steps for our class. So moving forward, I'll just call them the 2 3 carbon pieces, right? And I'll refer to them that way on the exam. So those 2 3 carbon pieces move into the mitochondria. And there, those 2 3 carbon pieces are going to get chopped up some more, right? They're going to get chopped up into eventually carbon dioxide, right? The hydrogen will be removed and the carbon and the oxygen will be the waste product. Right? So again, this process is called the Krebs cycle, the chopping down of those 2 3 carbon pieces. It's also sometimes called the citric acid cycle. Right? So just to confuse biology students, it has 2 names. Right? In this case, we generally refer to this stage of cellular respiration as the energy harvesting stage, because you're breaking the bonds between those 2 3 carbon sugar pieces and harvesting all the energy that's stored there. And the way that you're going to store that energy is in 6 NADH, 2 more ATP, right? And check this out, 2 FADH2s. These are another energy storage molecule, right? So you're harvesting the energy from breaking the glucose down into its CO2, right? And then you're storing all that energy as NADH and FADH2. Good? Any questions? Citric acid? Yeah, so don't worry about the pyruvic acid being converted to the acetyl-CoA. I just put that in there in case you want to geek out. All right, so glycolysis is taking that 6 carbon glucose and breaking it in half. It happens in the cytoplasm. It yields 2 ATP, but it also required an investment of 2 ATP. Right? Those 2 3 carbon sugars are going to be modified and then transported into the mitochondria, where they will run through the Krebs cycle. The Krebs cycle takes those 3 carbon pieces and chops them up to individual carbon pieces, right? And pulls the hydrogen off and stores it. The hydrogen and its electron goes onto NADH, right? So NADH is holding or taking the hydrogen that was once upon a time on the sugar, right? Holding its electron and the hydrogen. Later on, we're going to pull these off to run the electron transport chain. Same thing with FADH2, right? It's holding the electron and the hydrogen that was taken from the sugar, right? And it will be used in the electron transport chain. Alrighty, so the electron transport chain we already talked about, right? Now we're going to add this detail of, it's embedded in the cristae, the phospholipid folds inside of the mitochondria, right? So now we can imagine we're looking at cristae folds, right? And here comes our NADH that we harvested 2 in glycolysis. We harvested 6 during Krebs cycle. Now we're going to take the hydrogen off of that NADH, right? The electron is going to get ripped off and transported down the electron transport chain, and the hydrogen, right, is going to get pumped to the one side of the membrane to store energy. Yes? We're going to use, so the animation I think only showed NADH coming in here, right? NADH, kind of. FADH2, I'm not going to talk too much about. It's also temporary energy storage molecule. It has a different electronegativity, so it's just going to add its electron at a different location on the electron transport chain. Got a different energy potential. But again, don't worry about that too much. Just know that they're both contributing hydrogen and electrons to run the electron transport chain. Okay. Good. And again, the net goal of the electron transport chain is to create that hydrogen gradient storing energy. Can I go on? All right. Oh, yeah, that's why I put this picture in here. So here you can see NADH donates its electron and hydrogen over here. FADH just donates a little bit farther down the electron transport chain. They're just slightly different energy storage molecules. All right, and then your last step, chemiosmosis or oxidative phosphorylation, is taking advantage and using that potential energy that was stored in the hydrogen gradient. You're going to open the ATP synthase when you're ready to cycle ATP. Hydrogen ions are going to run down their concentration gradient using diffusion, right, from high concentration to low concentration. As they move through that ATP synthase, they literally turn it like a turbine, right? And that kinetic energy we can use to put a phosphate back onto an ADP, right? This is going to be the big yielding stage, right? We're going to yield 34 ATP from this stage. Wow. Right. 34 ATP, and this is also where oxygen at the end of the electron transport chain is going to grab the electron finally, and then turn around and bind with hydrogen ions that are in the environment to make water. Yeah, nothing's created, nothing's destroyed, it's just all reassembled. Any questions? Cellular respiration. So I have a worksheet that I'm going to load onto Canvas for you guys to review. It basically is, I don't know, like a bubble flow chart that walks you through what are the 4 steps, right? How much ATP, how much NADH, how much FADH is made at each step, where they occur. Yeah? So it'll help you to recap cellular respiration. So I'm going to skip that for a second. Just to overview, right, the 4 steps. It started with glycolysis. You take a glucose that you got from your digestive system, delivered by your cardiovascular system, heads into your cells from the extracellular matrix, right? Inside of your cell, you're going to run glycolysis using enzymes. It's going to take 2 ATP. It's our energy investment stage. We're going to break that glucose in half and yield 2 3-carbon sugars, right, or pyruvic acid. It's going to yield 2 ATP, but again, you invested 2 ATP, so you have a net no ATP. It also yields 2 NADH. Right? That pyruvic acid is going to get transformed to make it permeable to the phospholipid membrane of the mitochondria. So here you can see it's still the 2 3-carbon sugar. Now we call it acetyl-CoA, right? And those 2 3-carbon sugars are going to each be run through the Krebs cycle, where they're going to get chopped. All their hydrogen is going to get removed and attached either to the NADH or the FADH2, right? So 6 NADH, 2 FADH2s. This is where you're going to yield carbon dioxide, right? You're going to then use these energy storage molecules, the 2 ATP synthesized there as well. Then all the NADH, all the FADH2 that you harvested during this process, you're going to use to run the electron transport chain. Each one of them will donate their hydrogen, right? The electron from the hydrogen will be run down the electron transport chain, right? The hydrogen that's liberated, the hydrogen cation, is going to get pumped to one side of the phospholipid membrane to store energy like water behind a dam, right? And then when you're ready, you're going to run the 4th step here that I added in, chemiosmosis or oxidative phosphorylation. This is when you're going to open that ATP synthase, right? And allow hydrogen to run down its concentration gradient, utilize that kinetic energy to cycle ATP. And this is the big bang step, right? During chemiosmosis, you yield about 34 ATP.
So that second release of 2-NADH. Which step does that belong to?
You know what? I didn't talk about it. Let's pretend it's not there.
Okay.
It's a step I'm not talking about. It's one of the steps I cut out. So remember, we're breaking it down into four phases. Again, if you go on to take cell bio, you'll learn every enzyme. You'll learn every little nitty gritty of what happens. For us, I just want to do an overview. And, mostly the reason that I add this level of detail is so that you get the idea that it's super complex, right? It's kind of an amazing process that happens. And I also like to drop this in because when I talk about nutrition later on in the semester, I talk about some of the foods that yield you, right? Why proper nutrition is important, because some of the foods that you're consuming are providing you the building blocks for building NADH. And I like for you guys to know how important that is, right? You can't cycle ATP without NADH. Does that make sense? All right. Okay. Any questions? Aerobic cellular respiration. Oh, I forgot to mention the water. I keep forgetting to mention the water for some reason. If we go back and take a look at the electron transport chain for a second. One second. Oh, this one doesn't show it. So at the end of the electron transport chain, once you're done playing hot potato with that electron, right, it's going to get picked up by oxygen. Right? So oxygen is kind of like at the end of the electron transport chain, waiting for finally getting its electron. Right? Once it gets the electron, it's got a negative charge. It's going to turn around and bind with some of the hydrogen cations that are in the environment to form water. Right? So the last step is forming water. Water important for pretty much every reaction we run. Okay. Any questions? Aerobic cellular respiration. Okay. Aerobic cellular respiration, remember, was not the first kind of cellular respiration to evolve on Earth, right? Remember that early Earth was anaerobic. There wasn't very much oxygen in the atmosphere. It didn't accumulate until photosynthesis was invented by cyanobacteria ancestors. Anaerobic processes were the first type of cellular respiration that were being used by early prokaryotes. The environment was anaerobic. Right? And when oxygen accumulated, most of the anaerobic bacteria went extinct. It was the first mass extinction that we saw on Earth, right? Because oxygen is corrosive. Remember, it's electronegative. It's going to run around oxidating molecules in the environment, including prokaryotic cells. It was pretty deadly. Right. So now on Earth, most of those anaerobic lineages died out. There are still some anaerobic areas of Earth. There's still some anaerobic habitats, and we still see anaerobic cellular respiration being used by some organisms depending on where they're living. Right? And in anaerobic cellular respiration, we're not using oxygen. Right? And they tend not to release as much or cycle as much ATP because remember, oxygen and its electronegative properties, when it steals an electron, it's a big boom release of energy, right? That's what's allowing us to harvest so much ATP energy from aerobic cellular respiration, right? Is that property of oxygen. So anaerobic respirations or fermentations don't yield as much energy because they're not using oxygen. So anywhere from 2 to like in the 20s ATP, depending on the type of anaerobic respiration that you're talking about. They're not going to, since they're not using oxygen, they're not generating water at the end of electron transport chain. So they're going to have different types of waste products depending on the anaerobic process. And I'm going to talk about that in a bit, right? So not only do you have anaerobic prokaryotes living in, anaerobic conditions like mud flats or the geysers at Yellowstone, right? That are hot water, don't hold very much oxygen. They're anaerobic conditions. You're going to have anaerobic bacteria living there. You have, I think there's a couple there's a few unicellular eukaryotes that live in anaerobic environments that don't have aerobic cellular respiration, but it's not very common. Inside a multicellular organism, right, like us, we can sometimes experience anaerobic conditions in our cells. And in that case, we can switch to anaerobic pathways, right? So for us, for example, if you're exercising faster than your cardiovascular system can deliver oxygen to cycle ATP in your muscle cells, you can switch to an anaerobic pathway as a backup mechanism, right? So in our cells, we can use anaerobic pathways as a backup mechanism. And I'm going to talk a little bit about that lactic acid fermentation coming up. All right, so anaerobic pathways are oftentimes referred to as fermentations, right? The example that I'm just going to toss a couple examples so that you understand the diversity. There's all kinds of different fermentation pathways depending on what you're breaking down. Fermentation we use quite a bit in our everyday lives. It's a way that we can start to break down the starches in food products, right? So, for example, I'm going to talk about alcohol fermentation, what happens when you're making booze, right? So in booze, say you're making wine, right? Those grapes have quite a bit of sugar in them, right? That's why grape juice is so not healthy for you. It's got tons of sugar in it. So that grape juice that gets pressed, right, has all the sugar source. Then we add in potentially bacteria and yeast that are anaerobic, right? So yeast, just like you did in your little Whirly Pack experiments, gets added to that grape juice. And then you put your wine or your precursors to wine in an anaerobic vat. You put it in a tub where it doesn't have oxygen readily exchanging with the environment. And the yeast, which are unicellular eukaryotes, are going to switch to anaerobic pathways, right? The bacteria are going to switch to anaerobic pathways and break down that sugar, right? They're going to break it down. And in that process, they're going to yield carbon dioxide, kind of like aerobic cellular respiration. But instead of yielding water, they're going to yield ethanol, right? So the CO2 is what's going to make your wine potentially bubbly, right? If you're drinking a Lambrusco or beer, right? It's what makes beer bubbly, that carbon dioxide that's generated as a waste product from the yeast and bacteria. And the ethanol is the sugar that's been broken down into a different molecule, an ethanol, an alcohol molecule. Does that make sense? Fermentation, right? So instead of yielding water, you're yielding ethanol. Fermentation can result in all kinds of pathways. And again, remember that fermentations all are using glycolysis. Anaerobic pathways all use glycolysis as a first step. So here you can see that glucose broken down into pyruvate. And then we can ferment all kinds of different things using different either yeast species or bacteria species. So here we can take a look at some of our commonly fermented foods, right? And here are different types of yeast, right? So Saccharomyces is the yeast that we commonly use to make bread, right? When you're making bread, the sugar source is the flour, right? That you're mixing in. As you activate the yeast in those anaerobic conditions in the mass of dough that you mixed up, right? They're going to yield carbon dioxide, right? And that carbon dioxide is the gas bubbles that's going to cause your bread to rise, right? Or that dough to rise, anaerobic pathway. Same thing with making of soy sauce or of cheese. You're putting them in anaerobic conditions to force fermentation to happen. So yeasts are kind of interesting critters because they can use anaerobic or aerobic cellular respiration, and they'll switch to whatever pathway dependent on their environment. So if you were to, make your beer vat and it had a leak and air was getting in and oxygen was exchanging with the environment, the yeast would be like, screw fermentation. It only gets me 2 ATP. I'm going to run aerobic cellular respiration and yield 38 ATP. Huh? Right. Of course, the yeast is going to go for aerobic. And then your beer doesn't turn out, right? All right, in our cells, lactic acid fermentation is going to be using lactate in our cells and breaking it down into lactic acid. So again, this happens when you're in oxygen debt, right? And as your cells switch to anaerobic process and break down that sugar glycolysis and then convert it into lactic acid, right, to yield 2 ATP, right? This is a backup mechanism, right? Of course, you don't want to be cycling just 2 ATP when you could be using aerobic cellular respiration that cycles 36 to 38 ATP. Why would you do that? You're only going to use anaerobic in your cells when you are in oxygen debt. So in the event that you were on the savannah running away from a lion, right, aerobic cellular respiration is going to allow you to cycle a lot of ATP. If you are using more ATP than you can supply oxygen for, your backup mechanism is to short-term switch to anaerobic pathway lactic acid fermentation, which will maybe allow you to enough oomph to get away from that lion. You can't sustain that for a long time. That lactic acid actually starts to damage your cells. And this is why when you're working out and you switch to, your cells convert to anaerobic, that lactic acid is actually, what makes your muscles feel like they're burning, right? It's actually damaging your muscle cells. You shouldn't be, utilizing it long term. Does this make sense? Backup mechanism. And again, a lot less ATP. So I should mention, right, when we're talking about aerobic cellular respiration, we always say it yields 36 to 38 ATP. 36 to 38 ATP. The reason why, did you guys learn this in lab? It's accounting, right? You make, through all the steps of aerobic cellular respiration, 38 ATP. But remember, glycolysis is your energy investment stage. You invest 2 ATP. So if you subtract that out, you only yield 36. So depending on how you do your accounting, you might say you make 36 ATP to 38 ATP. It doesn't fluctuate. It's just how you count them. Yeah? Oh, I haven't done a poll in a long time. Let's see. Didn't do a good job of this today. All right, so I got easy breezy, fast poll. Why don't we, let's see if I can, today's 10:45. Why don't we see if I can finish photosynthesis in 5 minutes? So I'm going to move on unless anybody has questions about cellular respiration. We talked about aerobic, using oxygen, anaerobic, no use of oxygen, lots of different types of anaerobic. We just covered a few types. Yeah? Oh, I should have talked about kombucha as fermentation. That might be a fun one to add in. Your kombucha gets bubbly, yeah? And that's because the anaerobic little CO2 production. Ah, so fun. Biology, I love it. All right. So we spent a lot of time talking about cellular respiration. That's going to be our sample detailed metabolic pathway. For photosynthesis, I'm going to really just abbreviate it. I want you to understand that photosynthesis is important for life on Earth, but of the 10% of things that I want you to remember, the steps of photosynthesis and all the nitty-gritty detail are not the most important things that I want you to get out of this class. Cellular respiration applies to your body and your health, and so I want you to understand how that's significant to you. Don't tell Michelle, the botanist, if you take cell bio, or if you take oh yeah, she teaches us a lot, cell bio or botany. Don't tell her I dissed photosynthesis. Just say you were impressed at how amazing photosynthesis is, because it is pretty amazing. All right, so remember that photosynthesis is going to be taking the products of cellular respiration, that CO2 and water, and using sunlight energy to reassemble the sugar molecules by attaching 6 CO2s to each other and you're going to take the hydrogen off of the water and use it to build the sugar, and you're going to yield as a byproduct that leftover oxygen, right? So photosynthesis is going to yield organic molecules and oxygen. And don't forget, autotrophs are all going to be running photosynthesis, or virtually all autotrophs are going to be running photosynthesis. There are some deep sea autotrophs that are using other chemical pathways, for example. So plants, some eukaryotes, single-celled eukaryotes, like euglenids, are going to be photosynthetic. And then remember that bacteria, like cyanobacteria, can be photosynthetic as well. But the key difference here is that no membrane-bound organelles in prokaryotes, so they're just going to be running photosynthesis in their cells, not in a chloroplast. So for maybe I can't finish photosynthesis today, but let me see what I can do. All right, let's finish poll. Everybody voted? Last call? Poll?
Wait, that was for the ADP question, right?
Yeah.
Okay.
All right, so just to recap, your cycling of ATP during cellular respiration. Which of the following is the low energy state, ATP or ADP? Great job on that concept. Indeed, ADP is the low energy state, ATP is the high potential energy state. Make sure that you're understanding the cycling of ATP and the potential energy associated with those molecules.